program. And DNA is like hardware that is producing different proteins. Our classical knowledge shows that histones are found around the DNA, and in the 1990s we thought that histone proteins were around the DNA and they didn't have anything to do with familial passage of any disorder. When you go over the histones, then you see 3 billion letters. It's a huge molecule, but it is compacted 50,000 times. That's what we thought histones were, the compactors. You know that the genes first go through DNA and RNA, then there comes protein synthesis. I wanted to show you the difference between genetics and epigenetics. Here, genetics are explained by mutations. They're inherited. They're germline species, etc. Epigenetics is alterations which are more stable. We don't know. It could be a dynamic process, and we know that there's a lot of variability, and it is found in the soma cells. Methylation, acetylation, and modification, histone modifications. These are some of the concepts that we talk about. So when you look at the concept of epigenetics, you see that after fertilization, differentiation starts. There are more than 200 types of cells. The liver cells only have liver cell functions, for example, and brain cells are differentiated for cognitive functions. So all the cells are differentiated for different functions in the body. That's one way of expressing it. And the liver cells do not take part in cognitive processes, and vice versa. Brain cells don't have anything to do with liver. Epigenetic mechanism. In the DNA, the epigenetic mechanisms define some genes. Some genes are silenced and other genes are more expressed more effectively. And they have the possibility of stopping undesirable genes. DNA methylation is a process, and during this process the methyl groups make the undesirable genes into unfunctional genes. A human being will have healthy human epigenomics and unhealthy genomics as well. Genes are under the impact of radioactivity, chemicals, toxic materials. Epigenome is lighter, it's not toxic, it has normal and required physiological characteristics. Hormones, mediators, and environmental conditions and chemical factors may have positive or negative impact. And this effect will continue in the coming generations. So it is inherited, and histones also carry genetic materials. It's a dynamic process. This is only a summary of the previous slide. We know that characteristic expression of gene is very complex. There is a very complex pattern. There are about 23,000 genes, and they're all coded with 100 different proteins. They're all synthesized, so the whole thing is very complex. Environmental factors may have an impact on epigenetic mechanisms. In fact, we know that there is a million transformation. Maybe we are studying, so that they're coming up with different results. I believe that in the future we will be able to explain the situation. If you're interested in epigenetics, I'm sure you realize that, especially in cancer research, in the past 5 years, there are many new studies, technology is developing, and And all the publications are from 2004, 2005, 2006. So there's a lot of work going on in this subject. Epigenetics is very important for neuronal plasticity, learning, stress, adaptation to stress, and response to psychotropic drugs. Epigenetic mechanisms, you can see what I've already told you before. After fertilization, there is a demethylation in the genome of the father, and histone modification starts. There is also a step-by-step demethylation in the genome of the mother. The genes get together, and a different epigenomic model is created, which will belong to the baby. And here certain genes are silenced. Certain genes modify their activity. A new epigenetic model is born, and it starts with fertilization. This is a very popular slide. It's, as you can see, it summarizes everything, epigenetic mechanisms, and the factors which influence this process, intrauterine period, early childhood, environmental factors, aging, diet. Everything has an effect on the chromosomes. Here you see the chromatin. There are DNA and histone proteins, and here you see the methyl group. If the methyl group, when it binds, here you can see that DNA is not open and it cannot be synthesized. If there's no methylation, DNA cannot be active, gene cannot be active. Epigenetic factors that you see here, it could be a methyl group or it could be something else. That's when the synthesis starts. It seems like a simple mechanism, but actually it's highly complex. Thousands of molecules are involved. In my opinion, the slide is an excellent summary. This is very important for cancer, for autoimmune diseases, psychiatric disorders, and diabetes. DNA methylation. I spoke about three mechanisms. Gene expression is suppressed, embryonic development, transcription, chromatin structure. DNA plus histone proteins create euchromatin, heterochromatin. Euchromatin is more condensed, and its genetic expression is not so strong because there are inactive genes. X chromosome inactivation, genomic imprinting regulation. This is also very important. You will hear about this in the future. It will be quite popular as a subject. The alleles from the mother and father may carry some complications. At least one of the alleles may have an effect that is called imprinting. Certain genes are silenced at the beginning. The gene is suppressed, it's silenced, so there are two alleles. One of them is silent, and it will play a role in cancer formation, psychiatric diseases, etc. And there are many studies going on the subject. DNA methylation is indeed a highly important and complex procedure. DNA methyltransferase, DNA CpG regions. There are islets. That is where cytosine is methylated. That is part of epigenetic mechanism. In the genome there are transposons and also repeat sequences. Transposons are free DNAs. Heterochromatin is not highly condensed. It's in the CpG region. And methylation is quite high, and we see that transcription is suppressed and transposons are not active within the gene, and chromosome is highly active. DNA methylation, here we don't know where DNA methylation occurs. It all depends on the tissue and the aging process. In different parts of the brain there are different levels of methylation. Active genes mean low methylation. DNA methyltransferase, there are 5 enzymes. DNMT1, DNMT3L is not very active. I know this is very technical. Maybe I should skip over this part. It's a very small molecule. But I think in the future you will learn more about that small molecule. How many minutes do I have, Mr. Chairman? 2 minutes. Depression model in adult rats. There is a BDNF gene expression modification in the hippocampus. DNA methyltransferase is increased, and relay methylation is also increased. All these are new publications. The same study also shows that an increased anxiety where the mother does not take care of the baby rat. Here in the hippocampus, glucocorticoid receptor expression is decreased. I will skip over these parts. It's a very new publication. Childhood abuse or childhood neglect cases who later died because of suicide. They were compared to patients who did not have abuse or neglect stories, and the two groups were compared, and it was seen that there is a relationship between methylation, there is hypermethylation, and it shows that cytosine has an impact on methylation. We believe that research will continue in the future on the subject. Just 1 minute, Mr. Chairman. In schizophrenia, for example, DNA methylation could also be very important. Here you see an expression of DNA methylation in a normal patient and in a schizophrenic patient. There is a decrease in epigenetic process, the first attack, remission, progression of the disease, etc. Histone modification, that is also very important. DNA packaging, replication, and gene expression are very important in histone modification. There are two enzymes, histone acetyltransferase, and it frees the histone, and there is a gene expression and protein activity. And there is RNA-induced silencing, non-coded RNA, non-coding RNA, microRNA, small interfering RNA, X chromosome inactivation RNA. All these are like activating powers. They have very small RNA. They seem to be independent, but some people believe that there is a control mechanism. Thank you.